Oh.
Good to go? Yep. All right. So, thank you everyone for coming today. I am very happy to introduce Catherine Barnier, who is a grad student in my lab, who's going to be giving today's Tools and Tech seminar. Catherine got her bachelor's degree in chemistry at Western Connecticut State University. While she had been there, she did a bunch of research both at a lab at that university and also did an REU at the University of Southern Mississippi. She also picked up a bunch of awards, including things like an ACS Analytical Chemistry Award, the Outstanding Graduating Senior Award, good stuff. And so since coming to Michigan and joining my lab, she's been working on the project that she's going to talk about today, which is using crosslinking-based mass spectrometry to identify protein-protein interactions and their components.
Thanks for the introduction, Peter. Also, thank you everyone for showing up and giving me your time. Like Peter mentioned, I'm going to be talking about how we're analyzing some data from crosslinking-based mass spectrometry analysis, and definitely feel free to ask me questions as I go through, but I'm just going to give you guys a heads up. I'm going to try and end a little bit early because I promised Steve I wouldn't make him late for 5:27 right after this.
Promise.
Yes, I did promise, and I don't want to be heckled, so I'll do my best. Am I not advancing? I'm no longer advancing. My career is over.
What a metaphor for time for a new project.
What it is. Everyone enjoy the pizza. Have a nice day. Can I use just the keyboard? Is that?
Keyboard does work.
It does?
Yeah, but.
Steal the whole keyboard. This isn't, if it's supposed to advance with arrow keys, that's not working. Here you go. I'll just draw a protein-protein interactome for you guys.
You draw a DC4 off of that, right?
Probably, actually.
Now it's working.
It's working now?
This is working.
I have a regular laser pointer if needed, so if that one's not. Oh, okay.
I don't know what USB was.
Cool, nice. Okay, take two. So, protein-protein interactions are a huge part of biology. They play a role in basically, I don't know, take a cellular function, any cellular function, and you're going to run into a protein-protein interaction of interest. These can be very complex, ranging from just the interactome of a single protein domain, and then if you scale up to the whole bacterial cell or the human proteome, there's this huge wealth of data that's contained within protein-protein interaction networks, and we'd like to find a technique and a tool that will help us analyze this data in a high-throughput, automated way. Oh, I should mention before I go into this that this tool is very much still under development. So there is kind of a biology-heavy intro. So definitely interrupt me if there are any questions there. And I'll be talking a little bit about what we've done so far, but then also kind of fleshing out what we're planning to do after we collect more of this data. All right, so just to give you a sense, if you don't work in proteomics, where we kind of are in terms of our current limitations and the current state of the field. On the experimental side, we do have tools to probe protein-protein interactions, such as co-immunoprecipitation, where if you take a cell lysate and you incubate it with an antibody, you can, in theory, that antibody will bind to your protein, and you can purify out not only your protein of interest, but also all of its interacting partners. Some of the issues with this, well, the primary one is that it's low throughput. You also wind up being very dependent upon the antibody, whether or not it binds very well and how efficient it is at actually pulling down what you're looking at. A couple other techniques we have are yeast-to hybrid screening or bacteria-to hybrid screening. This one does have the advantage over co-immunoprecipitation in that it can be high throughput. And the idea here is that you have a construct with a bait and a prey protein, and if they are interacting, you'll see transcription of a reporter gene. If they're not interacting, then you won't see that transcription. This still has two major limitations, though. One is that you're looking usually in non-native conditions unless the proteins you're studying are specifically like yeast nucleus proteins. Otherwise, you're expressing your proteins in yeast, and they're not really supposed to be there. So depending on what you're looking for, the fact that you're not in your protein's native conditions could mess up your analysis. Also, this technique has almost ridiculously high false discovery rates sometimes. When I started this project, Peter was like, oh yeah, we have this technique, we can use it, but it's got really high false discovery rates. Phil Andrews, who is a collaborator on this project, was like, oh yeah, it's got really high false discovery rates. And when I went into the literature, the fact that I found even one extreme example where the false discovery rate was around 70% is like kind of insane. And I don't know why that isn't a bigger issue. This technique is still used all the time. It was used as like the experimental method or validation for the human proteome graphic that I just showed on the previous slide. So it's not great. We would still like something that doesn't have these false discovery rates, which is why we've leaned towards cross-linking-based mass spectrometry, because that definitely has nowhere near the false discovery rates of like Y2H or B2H. Okay, on the computational side, we're taking data from mass spectrometers, and then the output isn't actually something you can read right away. It's one of these binary raw files, raw for the case of Thermo Fisher, but all of them are some kind of binary file that you can then convert to some sort of open readable format. But in order to do this, you need a conversion library, and these are proprietary. They're distributed by the manufacturers of the instruments. And until the past few years, they have all only worked in Windows operating systems, which is intensely frustrating if you're trying to build a Linux-based pipeline. In the past few years, Thermo Fisher has kind of seen the light, and they released a Linux-friendly conversion library, and I think also Brucker did back in 2018. I think you're nodding your head. All right.
I just know about the Windows and then.
Yeah, and so that's definitely been something that's kind of held us back as developers trying to build proteomic pipelines, because we're not really able to reach that same high throughput or automated level that we have in like transcriptomics or genomics, because we're held back computationally by some of the stuff that the manufacturers put in place to begin with. Another one of these, and this is kind of a bad thing, kind of a pet peeve, and kind of just a sad truth, which is that proteomics has a heavy bent towards graphic user interfaces. And I totally understand, like, you want to know your audience. A lot of times these softwares are being used by people who are not computational, and so they're not comfortable working in a command line. The only problem that I personally have with that, given that I'm trying to build a pipeline, is that sometimes these tools then have no command line interface that's accessible, or it's very, very difficult to access a command line interface. And so as a result, trying to actually pull everything together into a unified pipeline has been a little bit of a piece of work, and I'm still working on it. So I'd still say these are probably the two main things that are kind of holding us back in terms of proteomics. So just to give you guys a sense of some of the questions we're trying to answer, or really, I guess, the bigger motivation for this project, just like why we're doing this in the first place. Obviously, we want to know what proteins are interacting, but that's kind of the question that everyone's trying to address. The things that we're trying to improve on is that we want to be able to address which proteins are interacting, but under controlled biological conditions. We don't want to have to express in yeast if we don't want to. We want to be able to do this in our system of interest. We also want to be able to use a high throughput technique that doesn't have false discovery rates that are astronomical. And then also, we'd like to have an automated analysis pipeline. My dream of dreams right now is to just have one config file that I have to put all this information into, and then I don't have to touch anything, and it will take the raw data and give me this beautiful output, no clicking required. This is also a question that I'm particularly interested in, and so I'll talk about it quite a bit at the end, which is the fact that cross-linking-based mass spectrometry data isn't just protein interaction data, it's actually low-resolution structural information as well, because these proteins have to be within a certain distance of each other in order for the cross-links to even physically cross-link. And so we can, in theory, use that to infer some of the quaternary structures of interacting proteins, regardless of whether or not we have a crystal structure of that. And then, of course, the big question that we always need to address is what are the biological implications of these interactions? And so to get into this a little bit, I want to introduce our model organism that we've been focusing on for this project and for the optimization and development of this tool. And basically anyone, I guess, who's heard any talk ever by anyone in the Fredolina lab knows that we really like to talk about E. coli. And this talk is no different. Our model organism. Oh no, it got cut off. This is supposed to say the adorable E. coli was provided thanks to Chengson. He made this and now I put it in basically every single talk. Thank you so much, Chengson. I'm sure you glad I brought that up. So thank you, Peter, for pointing that out. So we're working specifically with E. coli UTI-89, where UTI stands for urinary tract infections, which is indicative of the fact that this is a uropathogenic strain of E. coli. In fact, E. coli strains in general are the most prevalent causative agent of urinary tract infections by far. UTIs have annual incidence rates in women of 10%, men of 2%, and it doesn't really discriminate across different age groups or demographics. Everyone's getting UTIs, which results in a pretty large burden on the health industry. It costs $2.5 billion per year, approximately 1 million days of hospitalization, and about 13,000 deaths, and that's just in the US alone. And if all of those statistics aren't scary enough to convince you this is a pretty good model organism to be looking at, the World Health Organization back in 2017 actually published a list of priority pathogens, and in that top category, their priority one, most need to be studied right now pathogens, the E. coli family is front and center. So we're really interested in studying this and using this to develop our tool. And one particular thing to note about all of these critical pathogens, these are usually opportunistic pathogens that are hospital acquired. They're going after people whose immune systems are compromised. They're multi-drug resistant, and most interestingly, at least to me for this project, is all of these are biofilm producing. And biofilms I'm going to talk about just a little bit because they impart protective capabilities to bacterial populations. They're a big part of what makes them multi-drug resistant or drug resistant. And so this is just a little schematic of the formation of a biofilm. You have free-swimming cells that find a surface that they like. They decide to stick to it and start growing there. And then it develops into this pretty complex biofilm structure, which is made up of this whole system here. And biofilms, I mean, depending on the species that you're dealing with, you just have multiple subpopulations that are doing different subsets of regulation. And right now, we don't have a way to separate every single subpopulation in UTI-89, so we're focusing on the two that we are able to separate just through like a physical washout, which is this matrix subpopulation, which is the external kind of film-like portion of the biofilm. This has a lot of the structural components, the coralli, that provide cell-to-cell adhesion and also cell-to-surface adhesion, so they kind of hold the population together and protect it. And this we're calling the matrix fraction or the matrix subpopulation. I'll probably refer to it both ways throughout the talk. And then we also have this washout fraction, which is underneath the matrix, and these are more loosely associated free-swimming cells. These have flagellar components, whereas the ones in the matrix typically don't have any at all. And this actually makes up the majority of the biofilm mass. Okay, so before I actually go through that, does anyone have any questions on this background portion on the model organism that we're looking at? No? Okay.
I can just put in one plug here.
Yeah.
I think it's worth thinking about this as a very simple developmental process, because really you're getting effectively differentiation of populations. Mm-hmm. And that's important because, it's a reminder that all the methods you're talking about now, you're applying them to UTI-89, but they should be equally applicable to almost any biological.
Yeah, any system you can get the cross-linker into, you should be able to do this.
Which includes humans.
Yes. Mm-hmm. Okay. So my goal for the rest of this talk is to just walk through our overall workflow for this technique, which will involve both the experimental side and the computational side. So for starters, we have our UTI-89 cells that are growing on a plate. And if this is our crosslinker, it's called DC4, and I'll brag on its wonderful properties a little bit later in the presentation. You incubate the cells with this crosslinker, and it permeates the cell membrane and cross-links proteins that are interacting together. And what's really nice about this is that you don't have to lyse the cells beforehand like you do in co-immunoprecipitation. So you're able to, in theory, preserve cross-linking or preserve protein interactions that are maybe, what I'm looking for, they're transient or they're weak, that you otherwise might miss with something like co-immunoprecipitation. Okay, so then after we cross-link, I got these photos from Janet Price over in the Chapman lab. She's done the vast majority, if not all, of the handling protocols for the UTI-89 cells, preparing them for sample preparation for mass spec. So this here, this little circular portion in the center of this picture is the biofilm, and you'll see it has these like little wrinkly structures to it, and that's due to all the structural components in this matrix subpopulation. And then this little weird like starfish looking thing around it is just some of the substrates. This is just two stops of the plates. And so if we take this and we just add some buffer to it, I'm going based off of Janet's description here. She truly just like gently kind of swishes the flask around. And what happens is this matrix part dissociates from the surface and sort of just rolls up like a carpet. And so that's what you're kind of seeing here. It's a little, I don't know, burrito biofilm or something like that. And so when this rolls up, it allows the washout cells to then go out into the buffer. And so then we wind up with two fractions, a matrix fraction and a washout fraction that contain our two separate subpopulations that we're interested in looking at. All right, so after we have our two subpopulations, then we go into what is like the standard mass spec preparation, where we have to lyse the cells, purify the protein, and digest them so that we have these peptides that are small enough for mass spec. And then I think a lot of people in this room are comfortable enough with mass spec, but this is just a little diagram. If you have your sample, you ionize your sample so you have these charged peptides, and then you expose them to basically a giant electromagnetic field, which will separate them based on their ratio of their mass to their charge. They hit a detector, and then you get basically this mass spectrum. This is what, like, if you just Google mass spectrometry output, this is usually what shows up, where you have an intensity or a signal intensity for each one of these peptides. Then you can take that information and you can compare the fragmentation patterns that you see from your results to a database of known fragmentation patterns, and you can identify what proteins your peptides came from. That's the gist of mass spec. You can get fancier, though, by doing something like tandem mass spec, where if this first mass spectrometry is basically all of these steps up until here, then you get your initial mass spectrum, and then you can select some of these peptides or one of these peptides based on a bunch of different criteria. You can say, I only want peptides that have an intensity that's after a certain level because I want to make sure I have a lot of it. You can pull them based on a specific mass to charge ratio that you're looking for. You can do basically anything as long as you get creative enough. And if you're curious, you should talk to Manny in Phil Andrews' lab because he does this all day long. So you can select that peptide, and then you go through an additional fragmentation step. So it basically would be taking just one of these proteins or one of these peptides and fragmenting it further into even smaller pieces. You go through that whole process of separating them by mass to charge again, and you wind up with a second spectrum. And the reason this is important to note is because it's part of what we do in cross-linking-based mass spectrometry, because even if we have a cross-linker like I show here, we actually need to break it in order to be able to find it. And I'm going to walk through how that's done. So this is our little DC4 that I love so much. It makes my analysis downstream much simpler. This box here that's covering the center moiety, this is called a Dabco moiety, and it's 112 Daltons. And that's important because let's say this is a representation of our crosslinker. If it's crosslinking two peptides, and then we fragment it in that second step of the fragmentation, so like here, then what we get is basically four possible outcomes. So you can either have this Dabco moiety fragment and go over onto the blue peptide, or it can fragment and go over onto the green peptide. And it will only cleave at those two sites, or only fragment at those two sites. And that's really important because that's been one of the issues with cross-linking-based mass spectrometry for a while. The technique has been around for years, but people weren't able to use it very effectively because these cross-linkers either could not permeate the cell membranes to get in and cross-link the proteins in the first place, or they wouldn't fragment very well. And so the downstream analysis wasn't clean at all. You could miss the cross-links, or they could look like some other kind of modification, and it got really messy. This fragments super cleanly, and so what we're able to do in the mass spectra is just look for that shift of the Dabco moiety. So I'm going to kind of zoom in on this part for a second, because this is the gist of how we're actually finding the cross-links in the data. So here, if this is our peaks, our little intensities, and each one is associated with a different peptide fragment, then basically for this initial peptide, we'll have the blue peptide without the Dabco moiety, the blue peptide with it, and same thing for the green peptide. And because of that, any peptide that was cross-linked on one side will have this 112 shift. So we're able to pull out peptides that were cross-linked. We don't know what they were cross-linked to at first, but we're able to pull them out based on that 112 shift. So in this case, we would have peptide pair 1 would be AB, and peptide pair 2 would be C and D. In order to figure out what the interactions actually are, you have to go back to the peptide that they were fragmented from. And you basically just ask, if I add up the mass of these peptides, does it equal the mass of that original peptide? And so in this case, we would say, we would try and sum A and D, and if that equals the mass of A, then we would call that as an interaction. And that is like the key thing that we are looking for in all of this data, is to try and find where these crosslinks are overlapping. Then we can do the database searching just with those interacting peptides. And for that, my only requirement for the pipeline is that I wanted to have some kind of command line interface, and it has to be able to handle cross-link modifications. Right now, our collaborators recommended Andromeda, so that's what we've built in for now, but I'd like to expand it so that I can drop in pretty much any database searching program and use that based on what the user. Yeah.
When you do database searching, there's certain assumption. For example, you use trypsin cleavage. That means the N must be some amino acid. I wonder whether the cross-linked amino acids also has some requirement.
You mean like a way to, so I don't think you could say it has a primer so much as we know the crosslinks will only react with like primary amines. So typically you're going to see it at lysine residues. Is that kind of what you're thinking?
Yeah.
Right, but lysine residues, depending on their accessibility on the protein, they may or may not be cross-linked. So just looking for those isn't exactly enough to be able to pull them out.
So will it also link the N-terminal of the protein?
The end amino of...
The N-terminus.
Oh,
It will.
yeah, yeah.
Okay.
It definitely can, for sure. And those are usually probably going to be pretty accessible. Maybe, okay. Okay, so this is like a big part, so I just want to make sure, do we all follow how we identify crosslinks, or do we have any other questions? Cool. Right. Okay, so that allows us to identify which of the proteins are interacting, and that output is usually just like some kind of spreadsheet with, oh, you have this peptide and that peptide, and they're connected to these proteins. What we can do with that data, though, that's going to be a little bit more interesting, is we can build these interaction matrices where if along this x-axis, each column is a different protein, and then just take that axis and flip it so that each row is also that protein. So then along the diagonal, we would expect to see if this is like protein A, then we would expect to see some cross-links inside its own subunit, or within its own subunit, I should say. And then we would also expect to see some interactions with other subunits of other proteins. We do expect this matrix to be very sparse because you wouldn't expect every single protein to be interacting with every single other protein that's in the system. So right now we're actually collecting a lot more cross-linking-based mass spectrometry data. It's going to cost like a crazy amount, but it's going to give us the coverage we need to have maybe slightly less sparsity. Right now with our preliminary data sets, we don't really have enough coverage to do any statistics with any like real confidence, I don't think. So I'm just going to kind of talk through what we're hoping to do or what we're planning to do with the higher coverage data set once we've got it. So to model the data, most likely we'll have some kind of zero inflated distribution. And for anyone who's not familiar with zero inflated distributions, basically pick a distribution like a negative binomial and add a parameter that accounts for how often you will see zeros in your data. And that's kind of the simple version of what a zero inflated model is. So either a zero inflated negative binomial or a zero inflated Poisson would probably be a good fit for this data. But I don't want to like swear on either one of those until we actually see what it looks like. And then in terms of calling significant protein-protein interactions from this matrix, there's a bunch of ways that we could do it. Again, it's probably going to depend on the data. One way would just be to look for outliers, do an FDR correction, and calculate Q values from that. Another thing we could do that might be a little more interesting or maybe a little more robust would be to do a lasso. And then we would have a pairwise interaction term for basically each element in this matrix. And then because of the L1 shrinkage that lasso applies, it would push a lot of those parameters to zero, which in some data sets you wouldn't want because you're not expecting a lot of sparsity. But since we're actually expecting a lot of sparsity here, that might be a particularly appropriate method to use on this data.
Do you detect homodimers, the difference between homodimers and the self-interactions?
So it probably would depend on the cross-link distance. So for example, one of the ones that I'm going to show later, we are able to detect the difference, but it's just because either within the dimer versus across the dimer to the other side is like too far for the cross-link to actually reach. So you can detect it if you have the crystal structure then to map back to, but if you have no knowledge about what that quaternary structure looks like, probably not. Unless you built the model yourself, which I'm also going to talk about. Okay, so that brings us up through preparing the samples and then running them through mass spec. We identified the cross-links here, and we can get these interaction matrices from that. And then that allows us to call significant protein-protein interactions. And then now we get to the part that, like, I'm like super excited about this. Peter didn't mention this. Oh yeah, go ahead before I get into that part. Oh, because I made it randomly for the presentation. It might be symmetric. I don't know yet what the data will look like.
But theoretically, should it be symmetric or not?
Yeah.
It should be.
Yeah.
It should be exact.
Okay.
Okay. Oh, so any other questions other than critiques of my beginning?
This is the one I put.
No. Okay. Good. All right. This is the part that I really like because Peter, again, when he gave me my introduction, he didn't mention this. I'm not even sure if you know this, but a big part of why I joined your lab was because you absolutely just lured me in with the molecular dynamics. You let me do an MD project for my rotation, and it was just like the coolest thing in the world. I was straight out of a biochem degree, and I had no idea computational biology was like a thing at the time. I was just like, this is the coolest blend of like hardcore biochemistry and biophysics, and it's a beautiful thing. So I'm really excited about this next part of it because we get to use some molecular dynamics to take, again, this low-resolution structural information that we're getting from cross-linking-based mass spectrometry, and we can use that to infer the quaternary structures of complexes, even if we don't necessarily have, well, especially if we don't have the crystal structure for that complex. That's where it's going to be really valuable. Okay, so for example, one of the things that we looked at in our preliminary data was RNA polymerase crosslinks. And so for those of us who are unfamiliar with this, is a very, very toyish, cartoonish diagram of what RNA polymerase looks like. You have two alpha subunits
This
of
is bacterial RNA.
yes, E. coli. Sorry, E. coli RNA polymerase. Everything is E. coli, Peter. I had the little E. coli guy. So we have two alpha subunits, a beta subunit, and a beta prime, sigma and omega subunit. And in the data, we observed crosslinks between the beta and beta prime subunits, and also between the beta prime subunit and the omega, and beta prime and sigma. And what was really cool about this was when we took the known crystal structure for E. coli RNA polymerase, keeping in mind that the theoretical range for our crosslinker is about 6 to 30 angstroms, when we pulled the crystal structure and just mapped out where those crosslinks would be, all of them fell within that range beautifully, despite the fact that crystal structures have all of the crystal packing effects, and it's going to be slightly squashed out of what its comfortable, happy biological conformation would be. It worked beautifully. This was a good day when we saw this, and it's worked for other complexes as well. Peter did the same thing for all of the ribosomal proteins in E. coli, and every single one of the crosslinks, again, fell within a reasonable range and matched the crystal structures that we had. So this is really, really cool. So what I'm hoping to do with this, and what we've started to do with this, is we can use ensemble modeling weighted by the crosslink data. And the idea that if we have like a star protein and a triangle protein, the idea is we could just take the crystal structures of the individual proteins, or we could take like the I-taser predictions, which I've had to do for some of these. Thank you for those also. And you can just use something like I don't know how many of you are familiar with Rosetta. It's like a pose docking software, so we can just generate like 1,000 different possible conformations of these two proteins in relation to each other. And then from there, cluster them based on their similarity. And so if we have like conformation 1, for example, conformation 2 and conformation 3, and say in the data we observed two crosslinks between these different proteins, where crosslink A in black was observed, say, 20 times, and crosslink B in gray was only observed once, then the way that we would treat this would be to give the conformations that had only A lot more of the inferred weight based on the crosslink data. So like, for example, it might be 99.5% of the weight, and any of the ones that allow for crosslink B, even though we observed it so rarely that we wouldn't want to give it the same weight as something that mostly only had crosslink A. Okay, so I've actually started putting one of these systems together. And so in the data, we looked for crosslinks that were just kind of like interesting to us. And we came across PFLB, which is a pyruvate formate lyase enzyme. So it converts pyruvate to formate. And this is what it looks like. It has a dimer form. And just to bring this little diagram back, we saw intra-dimer crosslinks in both the matrix fraction and the washout subpopulation. So that part was everywhere. But the interesting part is that we also saw interactions between YFID and CatG, but only in the washout. So we're already seeing differential crosslinks even in our preliminary data. And so we want to investigate those and see what those quaternary structures actually look like. And so this is YFID, also known as GRCA, and then this is CatG. This one we took from crystal structure on PDB, but then these two were iTaser-generated structures. And so right now I'm equilibrating these structures. And so then once they like breathe a little bit, then we can put them into Rosetta and start generating all of those poses. And so I'm like just super excited about this part. It could be because this is like what I'm most recently actively working on. But so this is where we want to go with the quaternary. Yeah.
Rosetta is not for MD, so why you are equilibrating?
So just to get it out of the crystal structure. So for the iTaser ones, it might not be necessary, but for ones that are actually crystal structures, I definitely want to let it equilibrate so that it's more relaxed.
So when you run Rosetta, you make it rigid body docking or flexible docking?
That's a good question.
And also, if you are using Rosetta, you should not use MD to relax it. You should use Rosetta's own relax program so that the energy is compatible.
Right. So, okay. I see what you mean.
Well, I mean, but you're also using simulations for something else, right? I mean,
Right, yeah, and that's what I wasn't clear on. Because right now I'm
Stuff you
equilibrating
can
these
talk
with
about.
MD for other stuff that we're going to be doing with this. But for the part that ties directly into this part of the pipeline, yeah, we would use Rosetta Relax in order to relax those structures. Yeah, sorry, I'm setting up these simulations for NAMD runs right now, and so that was just on my brain.
Running MD for a long time.
Right, but like a long time. So I still think Rosetta is probably more efficient. Okay. And so that takes us through basically all of this workflow. Wow, 35 minutes. That was pretty impressive. And so does anyone have any questions on the overall workflow? Yeah.
Just thinking about moonlighting interactions and what do you think is the potential of your workflow to discover such interactions based off of to compare your data set with existing protein interaction data sets and discover moonlighting interactions which are
Find something new.
difficult to find otherwise?
Yeah, that's interesting. It would probably depend on like how often that was observed, whether it's a very transient interaction or it would just depend on how much it showed up in the data. I guess I wouldn't. I don't know. What do you think, Peter? I wouldn't expect us to necessarily be able to call it with confidence, because if it doesn't show up very much, it's already a pretty sparse data set.
Yeah, I guess it depends on what moonlighting means. Because people usually talk about moonlighting actions in the context of what we currently know.
Mhm.
Sometimes they turn out to be very important. So it's all about the frequency. If it occurs frequently and it's just not an interaction we expect,
Then we could find it.
then we can find it. I suspect that's what's going on with the PFLV stuff, because there's nothing annotated, for
Oh, yeah.
example, about CatG and PFLV.
Right, yeah,
interactions.
I totally
Very
forgot to
likely.
mention that.
And it's pretty interesting.
Yeah, so that was that's one thing I totally skipped over this, like what these actually are. So I mentioned that this is a pyruvate formate lyase, but this is activated by another protein called PFLA, which is just pyruvate formate lyase enzyme activating component. And YFID also can further activate PFLB, and it's estimated this adds like 18% of the total flux to that reaction then. But the thing that's kind of weird about CatG is that it's not related to that pathway at all, as far as I can tell. It's just like a scavenger for high concentrations of hydrogen peroxide. It runs after it and it finds it and then it breaks it down into water. So there's nothing that biologically makes it clear why this would be interacting with PFLB. That's a really good point. So yeah, so we might be finding something there.
It's nice, though, because it's heartening that you did get one interaction that is known.
Yeah.
PFLV with something else.
Okay. All right. Before I take any additional questions, I just got to say a bunch of thank yous. The folks in Phil Andrews' lab do all of our mass spec. Lolita Piersimoni in particular, she was an absolute rock. She just left to go to a new position in Germany, and I'm really sad to see her leave because she she's been absolutely essential to my understanding of this project. She answered every single question and would meet for coffee so many times just to walk me through diagrams in mass spec. Manny also did a lot of the mass spec design for their experiments. And then over in the Chapman lab, Matt Chapman has been a great found of knowledge on UTI 89, and Janet Price is also just absolutely awesome. I need to thank everyone in the Fredolina lab, especially the folks that showed up to my practice talk, and Richang gets a double thank you because she showed up to the practice talk and the actual talk. I need to thank the folks at everyone in the bioinformatics program, really, and Julia especially probably for keeping me mostly smiling fourth year, and I haven't lost my mind yet. And then Marcy, I'm just really grateful she gave me the opportunity to present at Tools and Tech in the first place. And I just need to acknowledge some funding. I was on the Proteome Informatics and Cancer Training Program, which was really a very good experience. If anyone's interested in more of the mass spec side of this, definitely talk to Alexi or consider taking his class, 551. It's really good. And then I'm also funded through a couple grants, an EBS Innovation Award and a NIAID grant that Peter has gotten. Okay. Any more questions? Yeah.
Okay. So you have verified in terms of quaternary structures whether your cross-linking data makes sense.
Mm-hmm.
But for the first question you want to ask, what other proteins that interact? There's not that many verification you have done.
So we've done some. I didn't really talk about it here. It's my backup slides on this system. So let me go back to my initial little biofilm slide. Okay. So we have looked just to do some biological verification of the mass spec and the cross-linking part. Is that what you're asking?
So to test whether the cross-linking data can correctly identify real interacting partners versus those that are not interacting. So you have verification of proteins that are known to interact whether the cross-linking distance is correct.
Yeah.
But you have not verified whether you introduce false positive EPI predictions for proteins that are not interact.
Okay, I think I understand what you mean. I'm not sure if we could determine what the false discovery rate would be like right now with the sparsity of the current data sets. That's something that we'll definitely have to consider once we get the full coverage stuff. I'm not sure how we would address that right now.
Well, I think so yes, you need the full data sets. You also need gold standards. We cross-reference the gold standards, and some ridiculously high percentage of the cross-links we get are known, but not all of them. And then there's a question, which of those are real and which of them aren't. I suspect in the end, we're going to have to spot check some randomly sampled subset. Because a lot of the current interactomes are built using.
Right.
So for any EPI project, the difficult part is not about positive. It's about negative set because people don't report it.
I didn't mention it actually, but going back, co-immunoprecipitation is sometimes referred to as a gold standard. You're already smiling a little bit. It's sometimes referred to as the gold standard for protein-protein interactions. If that is your opinion, my recommendation would actually just be to walk down the halls of MSRB3 and grab some graduate student in biochemistry or someone higher and ask them their opinion on it. The answer you will probably get is just that it is insanely easy to coerce things into co-expressing that don't actually interact. And so what would be the gold standard for protein-protein interactions? ITC?
Actually purifying them and doing ITC is a really good way to see if they've got a reasonable bind.
Or, for example, extracellular set proteins are not going to interact with intracellular proteins most of the time.
Mm-hmm.
So that could be one of the possible negative sets you can try to curate. So, but if reviewer asks you what are the new proteins interactions you can identify, then you need to have a benchmark result to show, given the cross-linking core, what is the probability that two proteins interact or not?
Yeah, for sure.
There's another nice method you call it out, it's fluorescence-based, that uses pair of fusion proteins, where you have one thing that acts as a base, tethers to one end of the cell, and then the other one acts as a prey and has a fluorescent protein attached to it. You can see if you induce polar localization of a non-polar protein, which works in a more high purified way.
So EIFC.
What?
EIFC.
Yes, that's right.
My understand. So what we find is even if just put the two pieces without linking any protein, just the fluorescence protein split in two, you can still see background.
No, this must be a different interaction because you're not splitting the fluorescent protein.
Okay, so you're just looking at whole localization by Fred.
Yes, yeah. Well, it's not even. It doesn't even need to be Fred. Actually, it's just microscopy, where one of the. So let's say you have a putative interaction of protein A with protein B. So you fuse protein A with a protein that has strong polar localization. Which means in a bacterial cell, it will get stuck way on one end of it. Then you fuse protein B with an intact fluorescent protein. And then you look for polar localization of the fluorescent protein that's attached to protein. And this has been shown with really good negative controls not to have the kind of noise that you get by splitting fluorescent proteins, for example, because there is no intrinsic affinity between the fluorescent protein and the polar localizer. And so this doesn't even require FRET. You just have to look at the cells under a fluorescence microscope and see if you've got polar localization. And unless protein B has intrinsic polar localization, which of course you control for by doing the same thing in cells that don't have the polar localized partner, you get a pretty good signal. So I suspect what we'll have to end up doing is something like taking a matrix of 30 or so proteins, some of which are supposed to interact and most of which are not, and run an assay like that and see what are both false positive and false negative.
Yeah.
Using this workflow, will you be able to detect the specific residues where the bindings
are? Where the binding between proteins, that could be inferred a little bit. But if you mean like the residues that are cross-linked, yes, that we can detect for sure. But that's not necessarily a direct indication that that's exactly where binding is happening. It means they're coming within a certain distance of each other, but a particular binding could be at an adjacent surface or somewhere nearby.
But then if we like use your scoring, like if it happens a lot more, then possibly that's the name.
I think I would just be careful with the use of the word binding site, just because I think that is a bigger question than just two amino acids that are within a certain distance. I don't know. Do you have an intuition on that?
Well, no, I think it's a good question and it's a good comment. It'd be great to. I think, Catherine, you're interpreting binding site to mean the residue is actually contributing to the binding free energy.
Yes.
That is not directly measured by this, right? But residues, it's certainly telling you what is near that interaction.
Correct. Yeah.
So it doesn't tell you anything about the biophysics that drives the interaction, but it definitely tells you what's near that interaction.
Any other questions? Cool. 5 minutes early and you're free.